As our world moves forward with time, many advancements and miraculous inventions have been invented and perfected over the years, especially in the medical field. Now, one brilliant invention in medical science is artificial blood. So today, I, Shaqil, together with my groupmates AIMAN, Adam, and AMIRUL, will be presenting our case study on artificial blood. Now, the objective of our case study is to answer two significant questions, which are: how do scientists make artificial blood, and how effective is it compared to the real thing? Artificial blood refers to a blood substitute or a product designed to mimic and perform some or all the functions of natural blood. It's primarily developed to address the critical shortage of donated blood for transfusions and medical treatments. Now, there are two types of artificial blood, which are hemoglobin-based oxygen carriers, also known as HBOCs, and perfluorocarbons, also known as PFCs. I said PFCs. It's phenomenal how far we've come in technology. To think that people used leeches for bloodletting 300 years ago.
So, speaking of the past, I think to better comprehend artificial blood, we must first dive deeper into its interesting history. The earliest record of artificial blood was in 1660, when William Harvey described how blood is separated. Not long after, some medical practitioners tried a range of such things, including beer, urine, milk, and sheep blood as blood separated. In the 1890s, the first attempts at creating blood substitute began using a solution, a milk-based mixture, but these were not viable alternative for transfusion. In the 1930s, efforts turned towards developing plasma-based solution as substitutes for blood volume expansion. In the 1940s, chlorofluorocarbon, which we will talk about later, emerged as potential candidates for artificial blood, but initial formulations faced challenges. In the 1970s, hemoglobin-based blood substitutes against tension. Scientists explored and modified hemoglobin molecule as oxygen carriers. However, early versions caused side effects, including high blood pressure and kidney damage. In the 1990s, several companies conducted clinical trials for various artificial blood products, including PFC-based solution and modified hemoglobin-based substitutes. However, safety concerns and efficiency issues led to setbacks, and many products failed to get approval. Currently, we can see that artificial blood has an ongoing area of research, but there still hasn't been a widely accepted and commercially available artificial blood substitutes for transfusion.
As mentioned before, there are two types of artificial bloods, HBOCs and PFCs. The production of HBOCs rely on synthesis of hemoglobin, the very same protein that can be found in our natural red blood cells. This protein is capable of carrying oxygen to our body tissue. Hemoglobin are made up of polyphenol rings that has a central iron atom which can bond to oxygen molecules. So, the question is, how do we obtain hemoglobin? Well, it happens so that a bacteria called E. coli is capable of producing human hemoglobin. So, the process starts with a sample of the bacteria, will be placed in a test tube to multiply. Nutrients will be provided to the sample in order for it to grow. Once the population is enough, the sample will be transferred to a seed tank, which is a big stainless steel kettle which acts as suitable environment for further growth. The seed tank will contain warm water, food, ammonia, amino acids, and vitamins, which are all necessary for hemoglobin production. When population gets larger, they will be moved to a fermentation tank, a bigger version of the seed tank. Once the amount of hemoglobin reaches its targeted value, isolation begins. The bacteria will be in centrifugal separator, which will isolate the hemoglobin using fractional distillation. Finally, the hemoglobin is transferred to a processing tank, where it will mix with water and electrolytes in order to become artificial blood. The production of PFCs, on the other hand, are completely man-made, in which it requires polymerization of hydrocarbon compound that mainly consists of fluorine and carbon atoms. However, hydrocarbons are insoluble in water. Therefore, the hydrocarbon must be emulsified in order to be administered into the veins in our circular system. Perfluorocarbon work by physically dissolving oxygen molecule in their liquid structure. When these liquids are exposed to an oxygen-rich environment, they absorb and carry oxygen.
Now, before we know whether if artificial or natural blood is better, we must first dive into when and why do we even need artificial blood in the first place. So, let's imagine this. A serious accident is caused nearby and there are many injured people and we lost tremendous amount of blood. Ambulance will drive quickly and arrive to try to save them, but due to the lack of natural blood types and each blood types that the patient requires, the paramedic will struggle to save them all. And this is where artificial blood comes in. The artificial blood does not require the blood donation from another patient because can produce a large scale from another factor. Additionally, artificial blood is designed to be compatible with the human body regardless of whatever the blood type is. A, B or O, it doesn't matter. Furthermore, artificial blood has a larger shelf life, 1 to 3 years compared to natural blood, which is 39 to 42 days. Besides that, artificial blood can lower the risk of infection during transfusion because the products used are sterilized. Therefore, the possibility of virus or disease transmitted are elevated. While on the other hand, the only drawbacks to using an artificial blood is that its only purpose is to transport oxygen through our body. That's it. While natural blood, on the other hand, is capable of doing that, while it is also having the benefits of being able to fight off pathogens in our body. With all that being said, we can generally conclude that in the case of a medical emergency, artificial blood can be far more superior than natural blood. That's it from me.
In conclusion, we have achieved our main objectives. We managed to understand how artificial blood is made, which is either by harvesting E. coli for HBOCs or by polymerization for PFCs. Furthermore, we explored how effective artificial blood is compared to natural blood. The study of artificial blood by scientists all over the world should continue in order to properly commercialize the product. This could be a step forward to humanity, as many lives in critical conditions could be saved once medical centers may properly use this revolutionary invention. These are the references used in our efforts for our case study. Thank you for watching.